I'm Hexin Chen. I'm going to give a talk. This title: How to Induce Inflammation Promotes Cancer Stem Cell Maintenance and Chemoresistance. Sorry. Oh, I cannot move my. Okay. So the inflammation has been well known to link to cancer. So this link has been established in 19th century by Rudolf Virchow. So inflammation can impact every step of cancer development, from cancer initiation all the way to cancer metastasis. And in the development tumor, we know the immune cells or the immune can play both the pro and anti-tumor tumorigenic roles. So immune cells can secrete many factors to affect tumor growth, tumor mutations, and so on. So that's there's a few interesting questions how the node cancer is unhealed wound. So the big question, how the immune inflammation is triggered from first place. So there are many reasons to trigger inflammation. For example, inflammation may be from diet, the environment factors, exposure. There may be the chemotherapy that can trigger inflammation. There may be the chronic inflammation, autoimmune disease, and also known the tumor cell can also trigger inflammation as well. So how this inflammation shape the tumor development is not well understood. So for a while, we know there are models to explain how the tumor initiates. So there are two models to explain the tumor initiation. I'm sure the first few speakers have introduced this concept. Now, one is called the statistical model. They believe the cancer is initiated from mutant cell which accumulate serious mutation, lead the cell to uncontrolled proliferation, eventually lead to tumor formation. During this process, the tumor cell continue to accumulate mutation, and then the cells interact with the environment that applies some pressure, continue to select its most aggressive tumor cells. And in this model, the immune cells play a very important role to select the most proliferative and fitter cells to proliferate. But also we are facing this new model. This new model, brought about in 1990s, the cancer stem cell model. Right now, they consider cancer is a kind of bad organ. There's only a small proportion of cell consider cancer stem cell. They can self-renew and deficient to different type of immune cells. Now, for the statistical model, for a while we think the immune cells just how to immune cells can play both the anti and the pro tumor genesis role. Now, you put an immune cell in this concept. There are two different models, and the immune cell play a different role here. Now we know the immune cells during the tumor development, the immune inflammation goes through different stages, from the inhibition stage, in equilibrium stage, and escape stage. So the first mutation will occur during the tumor development, and the immune cells may be able to recognize this mutant cell, and then both the innate immune cell and that immune cell can be triggered to eliminate these cancer cells, and then these cancer cells never become apparent observed. So this is called the intermediate stage. But when more mutation or epigenetic change in the cells, some cells maybe become more resistant, and then that's the immune cell can eliminate some cell, but some cell maybe kind of survive a little better. So the immune system and the tumor growth reach the kind of equilibrium, and this equilibrium can be a long time. And eventually, the tumor cell will escape from this immune surveillance and go up. Now we'll talk about this statistical model and the cancer stem cell model, how to explain the role of immune cells in these two models. Now for the statistical model, for a while, we think the immune system to promote tumor growth should introduce some mutations because the immune cell can secrete many cytokines to introduce mutations, and also immune cells can secrete many growth factors and we can promote tumor cell growth. That's why the immune cells, even though they can kill some cancer, at the same time can promote tumor growth. But if you put this immune system in a cancer stem cell model, how we're going to explain the immune system in a cancer stem cell growth or generation maintenance? So that's in order we'll understand the situation. That's how we try to answer some of these questions here. Now, thanks for the previous speaker, Dr. Paula introduced the breast cancer background. So we are using especially focus on the breast cancer stem cell, especially HER2-positive stem cell. So HER2 is oncogene belong to the human epidermal growth factor receptor family. It's overexpressed in 20 or 25% of breast cancer. So the breast cancer generally can be divided to, based on the cells market, can be divided to three major groups. So the ER positive is a hormone positive breast cancer, sometimes called a Numina A or Numina B cells. And then there's a HER2 positive breast cancer overexpressed or HER2 overexpression breast cancer cells. Another one is the triple negative, and there be ER negative, HER2 positive, HER2 negative, and PR negative, triple negative, sometimes called a basal type of breast cancer. So the HER2 type, even though only account for 20 to 25% of breast cancer, but this type of cancer is very aggressive. Even look at the survival curve here. So this is Numina A, Numina B. Basically, this is a hormone positive breast cancer. They have relatively better survival. But another two type of cancer here, the HER2 positive here, and also the basal, sometimes called triple negative, they're overlapping. So they have much worse survival rate compared to another subtype of breast cancer. So there are two different type of study all show similar pattern. So HER2 positive breast cancer is a very aggressive type of breast cancer. So we try to use this HER2 positive breast cancer model to study the relation between the inflammation and the cancer stem cell maintenance or generation. So we chose this model uses the MNT-free HER2 translated mouse model. So this mouse model, the overexpressed HER2 new in the mouse memory epithelial cells. 100% mice develop tumor eventually, but these mice have a long latency, about 200 days latency. So we are interested in how this HER2 drive the tumor development. Now, HER2, if you overexpress HER2 in vitro in a petri dish, you put it in the petri dish and grow this overexpressed gene in normal cells. They never grow tumor, never form tumor cells. But if you grow these mice, they're carrying the overexpression of HER2 in the mammary gland, these mice will develop tumor, but there's a long latency. So the conclusion here, that's HER2 overexpression by itself is not sufficient to transform normal cells into cancer cells. Something still need to help this transformation. So this put in the in vivo, that's what thought is environment contribute to this tumor development. So then question is what's the environment factor why these cells become tumor cells in vivo? So we choose a time point here before the tumor development. We get this memory tissues to analyze the gene expression pattern. One of the interesting we found is that a lot of inflammatory gene are expressed during this stage. At this pre-malignant stage. So inflammation at this stage is not as surprising because we know the cancer is also associated with inflammation. But the question is how the inflammation is triggered. So what's the driving force behind this inflammation? Now, the original thought just the tumor grow, the tumor going to die, and then the cells, all this content released from tumor cell will trigger the inflammation. Now, we will isolate this, when we see this cytokine expression, most likely we thought it's just, cytokines secreted by the immune cells. That's why we get a lot of cytokine expression in the tumor environment. But at this stage, we don't see obvious tumor yet, so it's just 4-6 months mice. Now, so we thought, well, wasn't it possible the cytokine can be secreted by the normal epithelial cells, not the immune cells? So we get a normal mammary gland, we get the transgenic mice and then white mice. We purify the normal epithelial cells and use this bead, and then we did this the gene expression file again. And interestingly, we found the mammary epithelial cells actually express this cytokine compared to this wild type epithelial cells. So this cytokine is not mainly contributed, from this stromal cell or the immune cells. Now, the information we know they can, affect the normal stem cell differentiation or self-renewal. So in normal situation, the stem cell put in the, the leash, the stem cell kind of proliferate is kind of inhibited or keep in a quiescent stage. So I'm wondering whether this information at this stage, how they affect stem cell activity in a normal mammary gland. So we analyze this mammary gland at this stage, we analyze the cellular composition. So the mammary gland is a well-studied study. They can have one single mammary stem cell, they can repopulate the whole mammary gland. So in the mammary gland composition at this stage, in the normal mammary gland, there are very small proportion of stem cell, only 0.8% cell stem cell. There are small portion of progenitor cell, they're called epithelial progenitor cell, and only 3.1%. But in this is a pre-malignant stage, about a 6-month stage. And then we found these HER2 transgenic mice have a large proportion of progenitor cells, but not expansion of the stem cells. And that gives we're wondering why this progenitor cell population expanded, not the stem cells. Okay, if the information going to promote proliferation of stem cell, we expect the stem cell population going to be expanded. But actually, we notice a progenitor population. So later, when we analyze the tumor from these mice, actually, the tumor shows exactly same phenotype as these progenitor cells. So not a stem cell population. So that will ask one question: why is the progenitor population, expanded, not a stem cell? Now there come to the stem cell, stem cell model. So the cancer stem cell model, they just possess their small proportion of cell have a stem cell like ability, but they don't tell you where is the origin of this stem cell. So this cancer stem cell can be origin from normal stem cell, can be origin from this progenitor cell, can also possible from deficient cells. Now we are seeing here, it's just a progenitor cell population expanded. So the question we're going to ask, whether this HER2 is overexpressed in the stem cell, and then promote stem cell to proliferate, and then somehow these cells, you can screw up, the division screw up, and then we get expansion of a stem cell. Or this is a HER2 overexpression expressed in the differentiated cells, and then they can trigger the differentiation. This cell going to regain this stem cell-like property. That's a question we don't know. But the interest here, we know here this is the progenitor cell population is expanded. To address this question, how does HER2 affect this is the property of these cells? So we use the more in vitro model to better, understand the potential mechanism. So in this case, we use the human normal epithelial cells. So we introduce these HER2 in these human epithelial cells is 10A cell, epithelial 10A cells. So we see whether these 10A cells, in these 10A cells, the HER2 can drive this differentiation or affect some property of these stem cells. So when overexpression is HER2 in these epithelial cells, we did the gene expression profile again. Interestingly, we see all this cytokine which is overexpressed in the transgenic mice also overexpressed in this HER2 overexpression the normal epithelial cells, epithelial 10A cells. So all the cytokine, CXCL1, CXL20, L1, CXCL8, L6, interferon and beta. And GCNS will show the inflammatory response gene upregulated, cytokine secretion pathway activated, interleukin-1 production, L6 signal pathway all are activated. So it's kind of a mimic what happened in vivo. So the next question, so how this cytokine affect the tumor cell growth or affect the stimulus? So the first thing we did was see how the cytokine affect the tumor growth, but actually none of the cytokine dramatically affect the tumor cell growth in vitro assay. So next we analyze whether the cytokine affect the stimulus or whether it can introduce the deficiency and then allow this cell to again this stimulus. So we did tumor sphere formation assay and then based on the genes profile, we patch all the cytokine chemokines to culture this cell in vitro. We found these three cytokines, L1 alpha, L1 beta, L6, and dramatically increase the tumor sphere formation ability. So based on here, control L1 alpha and beta 6. But even though these three cytokines can dramatically increase this tumor sphere formation ability, the L1 beta, even at the MR level, L1 beta dramatically upregulated, but at the protein level, we cannot detect this beta expression in the supernatant. So they don't only focus on this L1 and L6. So since these L1 assays can increase the stimulus, we want to know whether this happen in the normal, breast cancer cells. Whether this L1, L6 also required to maintain the cancer stem cells. So we use a CRISPR-Cas9 system to knock out L1, L6 in one of the HER2 positive breast cancer cell, HCC 1954. So and then we'll select this knockout cell line, and we use HER2 knockdown cell as a control, and then we end as a cancer stem cell population. When this L6 L1 is knocked down, the stem cell content dramatically decreased, dropped. So from this 80% dropped to maybe 0.5, 0.6%. So we did a lot of molecular work to characterize the single pathway, and it turns out HER2 triggers sequential activation of L1, L6 in these HER2 positive breast cancer cells. And then this sequential activation seems very important for the maintenance of cancer stem cells. Now, that's why I explain why knockout one of these gene also dramatically reduce the cancer stem cell contents when injecting this cell into the animal to further confirm this is really required to maintain the cancer stem cells. The parent cell can grow tumor very well, and the knockdown will knock out the cell line, can form a tumor, but efficiency is much lower, and also tumor grow much slower. So, and then the next question, what needs to happen in the primary breast cancer tissues? So we examine the IL-1 expression in the primary breast cancer tissue by tissue array. We find this is IL-1 indeed can be detected in the most many about 60 to 60% of breast cancer, and the HER2 IL-1 expression correlated with the HER2 expression status, and also HER2 IL-1 positive breast cancer most likely show this the cancer stem cell phenotype based on the CD44, CD24 phenotype. So if you're staining this breast cancer tissue with these two markers, some from breast cancer show this double positive phenotype, some show this most likely breast cancer phenotype, the CD44 positive, CD24 negative, some is more differentiated phenotype. So we see there's a correlation between the IL-1 expression and also cancer stem cell phenotype in the breast cancer tissue. So the next question asks if this is true, the IL-1 is very important for maintaining cancer stem cell, whether we can possible to target this pathway for cancer treatment. We use this is an inhibitor called IRAK inhibitor. IRAK is one of the downstream target for IL-1 single pathways. So we use a small chemical to block this IL-1 single pathway, and then also use a paclitaxel with a chemotherapy, and then we combine these two drugs together to treat the cancer as well. So this is the pre-treatment, the tumor formation, very similar in all these group of mice. After treatment, so this control treatment, the tumor continue to grow, and then use the IL-1 single pathway inhibitor has no dramatic effect on the tumor growth, seems slightly inhibit tumor growth, and chemotherapy drug definitely can inhibit tumor growth. Combination, we can see they have IL-1 inhibitor and also paclitaxel show some synergistic effect to inhibit tumor growth. And so inhibit cell growth is not surprising because you combine any two chemotherapy drug can inhibit tumor cell growth. And the problem it is for the typical chemotherapy drug, we know that chemotherapy drug can efficiently eliminate is deficient cancer cells, but it's not efficient to remove the stem cells. So to say whether this the IL-1 inhibitor can effectively inhibit proliferation of cancer stem cell, so we analyze the stem cell content after few cycle treatment. So we have combination treatment, we have this single treatment. So we analyze this cancer stem cell content after 21 days post-treatment. At this stage, you can see this cancer stem cell population of this here. So this is the control cells, about 1% cancer stem cells, and use this. This is, I think it's not 5.1. So yes, 5.1. So for the IL-1 inhibitor, they can reduce the cancer stem cell content a little bit from 5.1% to 5%. But interestingly, for the chemotherapy drug, we see this population expanded. So it's currently 11.8%. So the chemotherapy drug can eliminate this non-stem cell, but lead to enrichment of stem cell. But if you have a double treatment, this stem cell population can be reduced. Okay, this also shown this is in the quantitative data showing here. So and also we'll follow up after this treatment. So since this cancer stem treatment need to enrich the stem cell, we assume this after treatment, this is a chemotherapy triggered tumor will regrow very quickly. Indeed, as we show here, after treatment, we'll further follow up. This is a treated mice. For the chemotherapy treated mice, the tumor will recur very quickly and will regain the growth very quickly. But for the combination treated mice, you can see the tumor will maintain the same size for a relatively long time. follow after treatment, follow for another 20 days, the tumor stay at the same size, they don't grow. So this is the data showing the cancer stem cell target therapy not only can inhibit tumor growth, can also properly delay the tumor recurrence. So I'll go summarize the position I have so far. So overexpression of HER2 memory epithelial cell induce cell autonomous inflammation signaling promotes expansion of the new immunoprogenitor population at the pre-malignant stage. And HER2 induce sequential activation IL-1, IL-6 signaling pathway in breast cancer is critical for maintaining the cancer stem cell from conjugal blockade IL-1 signaling preferentially reduce cancer stem population, improve the chemotherapy efficacy, and delay the tumor recurrence. So far we can see this HER2, if you put this together, how this HER2 induce tumor development. We said the HER2 expression, there's a first mutation maybe induced promote proliferation or induce a differentiation to generation. This is the progenitor population, and then in these cells, and HER2 also induce this cytokine expression, help to maintain, to generate this stem cell population, to maintain the stem cell population. But one thing we are missing here, we're still not putting this in the in vivo, because these cytokines definitely going to affect recruited or activated immune cells. How the immune cells get involved in this process has not been understood right now. So that's a part of we're still working on right now. So I will thank the member who did this job. Most work was done by Dr. Xiu Liu, who is a postdoc in our lab, and also thank all the collaborators and then the foundation support. I'm going to stop here. Hopefully you have some questions.